Acquired weak D occurs due to the inheritance of capital C in the transposition to D, which results in a weakened expression of the Rhesus antigen. However, these individuals do not produce anti-D if exposed to Rhesus positive cells. R1, capital R1, little r genotypes have 10 to 15,000 antigens on their cells. Capital R1 is rare and has 15 to 20,000 per red cell, as there are 2 copies of the Rhesus gene. Capital R2 has 16 to 33,000 on the red cell. Rhesus deleted has about 100 to 200,000 antigens per red cell. The Rhesus D antigen is the most immunogenic of all protein antigens due to the quantity of the D antigen on the red cell surface, as well as capital C and capital E. It has been reported that as many as 80% of Rhesus negative individuals transfused with Rhesus positive red blood cells develop anti-D antibodies. The reason this type of blood is given is due to saving O negative and A negative blood for children and women of childbearing age. In major emergency situations, all men and women not of childbearing age would be given O positive blood. As many as 16% of Rhesus D negative mothers exposed to a fetal maternal hemorrhage from an Rh-D positive fetus will produce anti-D antibodies. This is a high percentage. Fetal maternal hemorrhage can happen, for example, due to traffic accident, and treatment involves giving prophylactic anti-D antibodies. This is a summary of the characteristics of anti-Rhesus antibodies. They are clinically significant as they can cause hemolytic disease of the newborn and hemolytic transfusion reactions. This summarizes information of anti-D antibodies. Notably, they can cause hemolytic disease of the newborn. Anti-small c antibodies are produced by individuals lacking the C antigen and can occasionally occur with anti-capital E. It is capable causing hemolytic disease of the newborn, though not as devastating as anti-D. It is still severe. Anti-capital C is produced by individuals lacking the capital C antigen. It is relatively rare and seldom causes hemolytic disease of the newborn. Anti-capital E is produced by individuals lacking the capital E antigen and may be naturally occurring, but seldom causes hemolytic disease of the newborn. Anti-capital E is the most common antibody in the UK. And finally, anti-small e antibodies occur in a capital R2 person and is weakly immunogenic, seldom causing hemolytic disease of the newborn.